Which of the following components of the initiation complex is required for the scanning of the mRNA to find the start codon in eukaryotic translation? AEIF2. BDIF4F CEIF3 DEIF1. Answer: BDIF4F. Explanation: The EIF4F complex, including EIF4E, helps to recognize and bind the 5' cap of the mRNA, allowing the ribosome to begin scanning for the start codon August. 3. CSIR Net Life Sciences 2018. In eukaryotes, the initiation complex involves the binding of the ribosomal small subunit to the mRNA's 5' cap. Which factor mediates this interaction? AEIF2. BDIF4E CEIF5 DEIF4A. DEIF4A. Answer: BDIF4E. Explanation: EIF4E is part of the EIF4 complex, which binds to the 5' cap of the mRNA to initiate translation in eukaryotic cells. Conclusion: the formation of the initiation complex is a highly regulated and crucial first step in protein synthesis. Understanding how the ribosome, mRNA, initiator TRNA, and initiation factors come together to form this complex is fundamental to mastering the process of translation. This detailed understanding is key for cracking the CSIR Net JRF Life Sciences exam. The formation of the initiation complex varies between prokaryotes and eukaryotes but remains central to the translation process. You said, initiation. Factors under regulation, ChatGPT said. Initiation factors under regulation. CSIR Net JRF Life Sciences, maximum detail, paragraph format, no tables, PYQs included with year and answers. The process of translation initiation in both prokaryotes and eukaryotes is a highly controlled and tightly regulated mechanism. Central to this process are the initiation factors, IFs, that facilitate the formation of the initiation complex, the first step in protein synthesis. These factors are involved in the assembly of the ribosome, the binding of mRNA, and the charging of TRNAs with their respective amino acids. In this context, the regulation of these factors ensures proper timing and efficiency of protein synthesis, responding to various cellular conditions. 1. Prokaryotic initiation factors. In prokaryotes, translation initiation is facilitated by three main initiation factors: IF1, IF2, and IF3. These factors help in the assembly of the ribosome, the binding of the mRNA, and the loading of the initiator TRNA. The specific roles of each factor are as follows. IF1, this factor binds to the A site of the 30S ribosomal subunit, preventing premature TRNA binding and promoting the correct positioning of the mRNA. IF2, this GTPase is responsible for binding the initiator TRNA, formylmethionine TRNA, and escorting it to the P site of the ribosome. It also facilitates the GTP hydrolysis that is necessary for the dissociation of the initiation factors once the ribosome is assembled. IF3, this factor prevents the premature association of the 30S and 50S subunits of the ribosome and helps stabilize the binding of mRNA to the small subunit 30S, aligning the mRNA correctly for translation initiation. These factors work in a concerted manner to assemble the initiation complex in prokaryotic cells and ensure accurate translation initiation at the start codon. 2. Eukaryotic initiation factors. In eukaryotes, the process of translation initiation is more complex and involves a larger number of initiation factors, known as AIFs. Key AIFs involved in eukaryotic translation initiation include AIF1, EIF1A. These factors help in the scanning process for the start codon and assist in the assembly of the initiation complex by binding to the small ribosomal subunit 40S. AIF2, this is a GTP binding protein that is responsible for the binding of the initiator TRNA, methionine TRNA, to the small ribosomal subunit. It also provides the energy required for the assembly of the initiation complex and for the recognition of the start codon. EIF3, this factor binds to the 40S ribosomal subunit and facilitates its interaction with the mRNA and initiator TRNA. EIF4F, this is a complex of initiation factors that binds to the 5' cap of the mRNA and helps in mRNA unwinding. It includes EIF4E, binds to the 5' cap of the mRNA, initiating the recognition of mRNA. EIF4G, links the 5' cap to the ribosome and interacts with EIF3 to form the initiation complex. EIF4A, a helicase that unwinds the mRNA to facilitate ribosome scanning. EIF5, this factor facilitates the assembly of the large ribosomal subunit 60S with the small subunit 40S and the initiator TRNA, marking the transition from initiation to elongation. These factors work together to ensure that the ribosome is properly assembled and the translation initiates at the correct start codon. 3. Regulation of initiation factors. The regulation of initiation factors is critical to the cell's ability to control protein synthesis in response to various environmental and cellular cues. Here are some key mechanisms through which initiation factors are regulated. In prokaryotes, phosphorylation of IF2. In response to stress conditions, the phosphorylation of IF2 can affect its function, potentially leading to a reduced rate of translation initiation. Regulation by regulatory proteins. In some cases, proteins such as regulatory RNAs or specific regulatory proteins, like riboswitches, can bind to the Shine-Dalgarno sequence of the mRNA, preventing the binding of the ribosome and inhibiting translation initiation. In eukaryotes, phosphorylation of EIF2. The phosphorylation of EIF2 is a major mechanism of regulating translation initiation in eukaryotes, especially under stress conditions. When EIF2 is phosphorylated, its ability to exchange GDP for GTP is impaired, leading to a decrease in the formation of the initiation complex and global inhibition of translation. This is a key regulatory mechanism during stress responses, such as in the unfolded protein response. EIF4E binding protein 4EBP. The activity of EIF4E, which binds to the 5' cap of the mRNA, is regulated by the binding of 4EBP. When 4EBP is phosphorylated, it releases EIF4E, allowing translation initiation to proceed. Under conditions of stress or low nutrient availability, 4EBP remains bound to EIF4E, preventing the initiation of translation. mTOR pathway. The mTOR mechanistic target of rapamycin pathway is a key regulator of protein synthesis. Under conditions of nutrient availability, mTOR activates EIF4E by phosphorylating 4EBP, promoting translation initiation. Conversely, under nutrient deprivation, mTOR is inhibited, leading to a decrease in translation initiation. Regulation by small RNAs. In eukaryotic cells, microRNAs can bind to the 3' untranslated region (UTR) of target mRNAs and prevent their translation by inhibiting the interaction with the initiation factors of the ribosome. 4. Role of initiation factors in translational control. The regulation of initiation factors not only ensures proper translation initiation but also plays a key role in cellular responses to various signals. Cellular stress. Stress conditions such as heat shock, oxidative stress, or nutrient deprivation can trigger the phosphorylation of initiation factors, leading to a global decrease in translation to conserve energy and resources. Developmental regulation. The selective activation or repression of translation initiation factors allows for the regulation of gene expression during different stages of development. Cancer and disease. Abnormal regulation of initiation factors, such as EIF2 and EIF4E, has been implicated in cancer and other diseases. Overexpression of EIF4E in cancer cells can lead to uncontrolled protein synthesis and tumor growth. Previous year questions. PYQs 1. CSIR Net Life Sciences 2018. Which of the following is not an eukaryotic translation initiation factor? AEIF1. BDIF2 CEIF3 DEIF5A. Answer: DEIF5A. Explanation: EIF5A is involved in elongation, not initiation. The other factors listed are involved in the initiation process. 2. CSIR Net Life Sciences 2019. In eukaryotic translation initiation, EIF2 is involved in the binding of the initiator TRNA. What is the regulatory role of EIF2 phosphorylation? It enhances the binding of EIF4E to the mRNA cap. B. It promotes the exchange of GTP for GTP in the initiation complex. C. It inhibits translation initiation under stress conditions. D. It facilitates the recruitment of the ribosomal large subunit. Answer: C. It inhibits translation initiation under stress conditions. Explanation. Phosphorylation of EIF2 leads to inhibition of its GTP exchange activity, reducing the formation of the initiation complex and thereby global inhibition of translation during stress responses. 3. CSIR Net Life Sciences 2020. Which of the following eukaryotic initiation factors binds to the 5' cap of the mRNA to initiate translation? A. EIF2. B. EIF3. C. EIF4. E. D. EIF4G. Answer. C. EIF4E. Explanation. EIF4E binds to the 5' cap of the mRNA and is essential for the initiation of translation in eukaryotes. Conclusion. The regulation of initiation factors is crucial for controlling protein synthesis in response to cellular conditions. In both prokaryotes and eukaryotes, initiation factors such as IF1, IF2, EIF2, and EIF4E are essential for the assembly of the initiation complex and the accurate and timely initiation of translation. Understanding the regulation of these factors is important for understanding cellular responses to stress, development, and disease. This knowledge is vital for performing well in the CSIR Net JRF Life Sciences exam. You said. Elongation and elongation factors. ChatGPT said. Elongation and elongation factors. CSIR Net JRF Life Sciences. Maximum detail. Paragraph format. No tables. PYQs included with year and answers. Elongation is a critical phase in protein synthesis, occurring after the initiation complex is formed and the ribosome is ready to translate the mRNA into a protein. During elongation, the ribosome moves along the mRNA in a 5' to 3' direction, reading each codon and using the corresponding turn to add the correct amino acid to the growing polypeptide chain. This process is facilitated by a set of proteins known as elongation factors, which play essential roles in ensuring the efficiency, accuracy, and regulation of translation elongation. 1. Elongation mechanism. The elongation cycle involves 3 major steps: amino acyl turn of binding, peptide bond formation, and translocation. Amino acyl turn of binding. The ribosome consists of 2 subunits: the large subunit 50S in prokaryotes, 60S in eukaryotes, and the small subunit 30S in prokaryotes, 40S in eukaryotes. The turn of carrying the correct amino acid enters the A site of the ribosome, a process facilitated by elongation factors. The elongation factor EF2 in prokaryotes or EF1A in eukaryotes binds to the amino acyl turn and escorts it to the ribosome. This factor ensures that the turn correctly matches the mRNA codon in the A site. The GTP bound to EF2 or EF1A provides energy for the correct binding of the turn, and GTP hydrolysis releases the factor once the turn is properly positioned in the ribosome. B. Peptide bond formation. Once the correct amino acyl turn is bound in the A site, the peptidyl transferase center of the ribosome catalyzes the formation of a peptide bond between the amino acid in the P site and the amino acid in the A site. The polypeptide chain grows by 1 amino acid. C. Translocation. After peptide bond formation, the ribosome undergoes a conformational change that shifts the turn from the A site to the P site, and the empty turn in the P site is moved to the E site for exit. This process is facilitated by elongation factors and ensures that the ribosome advances by 1 codon along the mRNA, preparing it for the next round of elongation. 2. Elongation factors in prokaryotes and eukaryotes. Elongation factors are essential for the efficiency and accuracy of translation elongation. Although the overall process is similar in both prokaryotes and eukaryotes, the specific elongation factors differ between these organisms. Prokaryotic elongation factors EF2. Elongation factor 2. EF2 is a GTP binding protein that binds to amino acyl turn and escorts it to the ribosome. The GTP bound to EF2 is hydrolyzed once the turn is in the A site, ensuring proper codon anticodon matching. This results in the dissociation of EF2 from the turn and ribosome. EFT's elongation factor Ts. EFT's functions as a GTP exchange factor for EF2, facilitating the replacement of GTP with GTP in EF2 so that it can participate in subsequent rounds of elongation. EFG elongation factor G. EFG is responsible for driving ribosomal translocation. After peptide bond formation, EFG binds to the ribosome and uses energy from GTP hydrolysis to move the ribosome along the mRNA by 1 codon. This allows the turn to shift from the A site to the P site, and the empty turn in the P site moves to the E site for exit. Eukaryotic elongation factors EF1A. Eukaryotic elongation factor 1A. EF1A is functionally similar to EF2 in prokaryotes. It binds to amino acyl turn and facilitates its entry into the A site of the ribosome. EF1A is a GTPase that ensures the correct turn is matched to the mRNA codon. GTP hydrolysis leads to the release of EF1A after turn of binding. EF1B. Eukaryotic elongation factor 1B. EF1B functions similarly to EFTs in prokaryotes by promoting the exchange of GTP for GTP on EF1A, enabling it to participate in the elongation cycle. EF2. Eukaryotic elongation factor 2. EF2 is responsible for the translocation of the ribosome along the mRNA after peptide bond formation. Similar to EFG in prokaryotes, EF2 binds to the ribosome and uses energy from GTP hydrolysis to move the ribosome by 1 codon. This allows the turns in the A and P sites to shift positions, making space for the next turn to enter the A site. 3. Regulation of elongation factors. The activity of elongation factors is regulated to ensure that protein synthesis proceeds efficiently and accurately under different cellular conditions. Phosphorylation of EF2. In eukaryotic cells, EF2 can be phosphorylated by EF2 kinase, which inhibits its activity and halts ribosomal translocation. This regulatory mechanism is especially important during stress conditions or when energy is scarce. Regulation of EF1A. The activity of EF1A can be regulated by GTPase activating proteins GAPs and guanine nucleotide exchange factors GEFs, which influence the exchange of GTP for GTP on EF1A, thus controlling the elongation process. Feedback regulation. The availability of turnus, amino acids, and GTP can influence the efficiency of translation. In the case of amino acid starvation, the cell reduces protein synthesis by inhibiting the activity of elongation factors. Previous year questions. PYQs 1. CSIR Net Life Sciences 2017. Which of the following elongation factors is responsible for ribosomal translocation in prokaryotes? A. EF2. B. EFT circuit EFG DIF3. Answer. C. EFG. Explanation. EFG is responsible for ribosomal translocation in prokaryotes after peptide bond formation. It uses GTP hydrolysis to move the ribosome along the mRNA. 2. CSIR Net Life Sciences 2019. Which of the following is true about EF2 in eukaryotic translation? A. It is involved in the binding of amino acyl turn to the A site. B. It catalyzes the formation of peptide bonds. C. It is responsible for ribosomal translocation along the mRNA. D. It is required for the dissociation of the ribosome after translation. Answer. C. It is responsible for ribosomal translocation along the mRNA. Explanation. EF2 is involved in the ribosomal translocation step, using GTP hydrolysis to move the ribosome along the mRNA after the peptide bond is formed. 3. CSIR Net Life Sciences 2020. Which prokaryotic elongation factor is responsible for the amino acyl turn of binding to the A site of the ribosome? A. EF2. B. EFG C. EFT DIF2. Answer. A. EF2. Explanation. EF2 binds to the amino acyl turn and escorts it to the A site of the ribosome, where it is checked for codon anticodon matching before peptide bond formation. Conclusion. The process of elongation and the action of elongation factors are essential for the synthesis of proteins in both prokaryotic and eukaryotic cells. Factors such as EF2 and EFG in prokaryotes, and EF1A and EF2 in eukaryotes ensure that protein synthesis occurs accurately and efficiently. Understanding these factors and their regulation is crucial for performing well in the CSIR Net JRF Life Sciences exam. You said. Termination. ChatGPT said. Termination of transcription and translation. CSIR Net JRF Life Sciences. Maximum detail. Paragraph format. No tables. PYQs included with year and answers. Termination is a vital phase in the processes of transcription and translation that ensures the proper conclusion of gene expression, leading to the release of newly synthesized RNA or protein. Both transcription and translation have distinct mechanisms of termination, each involving specific factors and sequences to signal the end of these processes. 1. Termination of transcription. Transcription is the process by which RNA is synthesized from a DNA template, leading to the production of mRNA, rRNA, and tRNA. The termination of transcription marks the end of RNA synthesis and occurs when the RNA polymerase encounters a termination signal. This is achieved by one of two mechanisms: intrinsic termination in prokaryotes or row-dependent termination also in prokaryotes, and polyadenylation-dependent termination in eukaryotes. Intrinsic termination prokaryotes. Intrinsic termination is also known as row-independent termination. It occurs when the RNA transcript contains a palindromic sequence that forms a hairpin loop followed by a series of uracil residues fuse. This loop disrupts the RNA polymerase's interaction with the DNA template, causing the enzyme to dissociate and release the newly synthesized RNA. The formation of the hairpin loop causes RNA polymerase to pause, and the weak RNA-DNA hybrid in the transcription bubble due to the U-rich sequence leads to the release of the transcript. Row-dependent termination prokaryotes. The Rho protein plays a critical role in row-dependent termination. This mechanism requires the Rho factor, a helicase that binds to the RNA at a Rho utilization site. It moves along the RNA transcript in a 5' to 3' direction using ATP hydrolysis. Once Rho reaches the RNA polymerase, it causes the polymerase to dissociate from the DNA, releasing the newly synthesized RNA. C. Termination in eukaryotes. In eukaryotes, transcription termination is often coupled with the process of RNA processing. The most common mechanism of termination in eukaryotic transcription is polyadenylation-dependent termination. The RNA polymerase II synthesizes a precursor mRNA that is then processed by enzymes that cleave the mRNA and add a poly A tail. The termination signal is a cleavage site in the mRNA, and after cleavage, the RNA polymerase continues to transcribe a few extra nucleotides, which are eventually released as the RNA polymerase dissociates from the DNA template. 2. Termination of translation. Termination of translation refers to the process by which a ribosome stops synthesizing a polypeptide chain once a stop codon is encountered. This process involves the recognition of specific stop codons, UAA, UAG, UGA release factors, and the dissociation of the ribosomal subunits. Stop codons and release factors. Stop codons are sequences in the mRNA that signal the end of translation. The most common stop codons are UAA, UAG, and UGA. In eukaryotes, release factors ERF, and in prokaryotes, RF1, RF2, and RF3 are proteins that bind to the ribosome when a stop codon is positioned in the A site of the ribosome. These release factors catalyze the hydrolysis of the bond between the polypeptide chain and the tRNA in the P site, leading to the release of the completed polypeptide. B. Mechanism of termination recognition of stop codons. When a stop codon enters the A site of the ribosome, the release factor binds to the A site, triggering the termination process. For eukaryotes, ERF is the main release factor, whereas in prokaryotes, RF1 recognizes UAA and UAG, and RF2 recognizes UAA and UGA are involved. Polypeptide release. The release factor catalyzes the hydrolysis of the bond between the polypeptide and the P site in the tRNA, releasing the newly synthesized protein from the ribosome. Ribosome dissociation. After the polypeptide is released, the ribosomal subunits dissociate from the mRNA. The small and large subunits of the ribosome separate, and the mRNA is released from the ribosome. This marks the end of translation. Post-termination events. In eukaryotes, the ribosome is disassembled by ABCD1 and other factors, and the mRNA may be recycled for another round of translation or degraded. In prokaryotes, the release of the polypeptide chain and ribosome disassembly is aided by additional factors like GTP hydrolysis by RF3. 3. Post-termination modifications. After the termination of translation, the newly synthesized polypeptide undergoes various post-translational modifications, such as folding, cleavage, and addition of functional groups e.g., phosphorylation, glycosylation to become fully functional. Previous year questions PYQs 1. CSIR Net Life Sciences 2018. Which of the following is the role of Rho factor in transcription termination in prokaryotes? A. It is responsible for the cleavage of the RNA transcript. B. It helps in the formation of the hairpin loop. C. It is a helicase that unwinds the RNA-DNA hybrid, causing RNA polymerase to dissociate. D. It catalyzes the formation of the peptide bond. Answer. C. It is a helicase that unwinds the RNA-DNA hybrid, causing RNA polymerase to dissociate. Explanation. Rho factor is a helicase that moves along the RNA and causes RNA polymerase to dissociate from the DNA template upon reaching the transcription terminator. 2. CSIR Net Life Sciences 2017. In eukaryotic translation termination, which release factor is responsible for recognizing the stop codon and triggering the release of the polypeptide chain? A. RF1. B. RF2. C. ERF-DEF1A. Answer. C. ERF. Explanation. ERF eukaryotic release factor is responsible for recognizing the stop codon in the A site of the ribosome and catalyzing the release of the polypeptide chain. 3. CSIR Net Life Sciences 2019. Which of the following is not involved in the termination of translation in prokaryotes? A. Release factors. B. Ribosomal subunit dissociation. C. Cleavage of the polypeptide chain. D. Polyadenylation. Answer. D. Polyadenylation. Explanation. Polyadenylation is a process that occurs during transcription in eukaryotes and is not involved in translation termination. Termination of translation involves release factors, ribosomal subunit dissociation, and polypeptide cleavage. Conclusion. Termination in both transcription and translation is crucial for the proper completion of gene expression. Understanding the mechanisms of termination, including the roles of Rho factor, release factors, and the signals that trigger these processes, is essential for mastering CSIR Net JRF Life Sciences. You said, genetic code, ChatGPT said, genetic code. CSIR Net JRF Life Sciences, maximum detail, paragraph format, no tables, PYQs included with year and answers. The genetic code is the system by which genetic information is stored in DNA and RNA and used to direct the synthesis of proteins. It is a fundamental set of rules that dictates how sequences of nucleotides in DNA or RNA are translated into sequences of amino acids in proteins. This code is universal, though there are slight variations in certain organisms. 1. Characteristics of the genetic code. A. Universality. The genetic code is nearly universal, meaning it is the same in nearly all organisms, from bacteria to humans. This is a key piece of evidence for the common origin of life. However, there are some exceptions in certain organelles, like mitochondria and some species. B. Redundancy. Degeneracy. The genetic code is degenerate, meaning multiple codons can code for the same amino acid. For example, the amino acid serine is encoded by UGU and UGC codons. This redundancy in the code helps reduce the effects of mutations, as a change in the third nucleotide of a codon often does not alter the amino acid encoded. C. Non-overlapping and comma-less. The genetic code is non-overlapping. Each nucleotide in a sequence is part of only one codon. Moreover, the code is comma-less, meaning there are no gaps or spaces between codons. D. Directionality. The reading of the genetic code is directional, always proceeding from the 5' to 3' direction on the mRNA strand. This 1. Characteristics of the genetic code. A. Universality. The genetic code is nearly universal, meaning it is the same in nearly all organisms, from bacteria to humans. This is a key piece of evidence for the common origin of life. However, there are some exceptions in certain organelles, like mitochondria and some species. B. Redundancy. Degeneracy. The genetic code is degenerate, meaning multiple codons can code for the same amino acid. For example, the amino acid serine is encoded by UGU and UGC codons. This redundancy in the code helps reduce the effects of mutations, as a change in the third nucleotide of a codon often does not alter the amino acid encoded. C. Non-overlapping and comma-less. The genetic code is non-overlapping. Each nucleotide in a sequence is part of only one codon. Moreover, the code is comma-less, meaning there are no gaps or spaces between codons. D. Directionality. The reading of the genetic code is directional, always proceeding from the 5' to 3' direction on the mRNA strand. This ensures that the correct sequence of amino acids is synthesized during translation. 2. Codons, the building blocks of the genetic code. The genetic code is written in triplets of nucleotides called codons. Each codon corresponds to one of the 20 standard amino acids, or it signals the end of translation. Stop codons. There are a total of 64 possible codons, 4 cubed, since there are 4 bases in RNA: A, U, G, C. Of these, 61 codons specify amino acids, while 3 codons serve as stop signals. A start codon. The start codon is always August, which codes for the amino acid methionine in eukaryotes and formal methionine in prokaryotes. This codon signals the beginning of translation. B stop codons. The stop codons are UAA, UAG, and UGA. These do not encode any amino acid but instead signal the termination of protein synthesis, causing the ribosome to release the newly synthesized polypeptide chain. 3. The triplet nature of the genetic code. Each amino acid is encoded by a sequence of 3 nucleotides, known as a codon. For example, the codon UUU codes for the amino acid phenylalanine, while August codes for methionine. The sequence of codons in the mRNA determines the sequence of amino acids in a protein, which ultimately determines the protein structure and function. 4. Features of the genetic code A start codon. The start codon August not only signals the beginning of translation but also sets the reading frame for the ribosome. This ensures that the correct codons are read in the proper order during protein synthesis. B stop codons. The 3 stop codons UAA, UAG, and UGA signal the termination of translation. These codons are recognized by release factors, which facilitate the release of the polypeptide from the ribosome. C redundancy. As mentioned, the genetic code is redundant. For example, the amino acid leucine is encoded by the codons UUA, UUG, CUU, CUC, CUA, and CUG. This redundancy helps minimize the impact of mutations, as changes in the third position of a codon often do not result in a change in the encoded amino acid known as silent mutations. D universal code. The universality of the genetic code across most organisms is an essential concept in molecular biology. It implies that the machinery for protein synthesis is fundamentally the same in all living organisms, providing evidence for the common evolutionary origin of life. 5. Exceptions to the genetic code. While the genetic code is almost universal, there are a few exceptions. In mitochondria, the genetic code differs slightly. For example, in human mitochondria, the codon UGA, which is a stop codon in the standard code, codes for tryptophan. Some protozoans also exhibit variations in their mitochondrial genetic code. 6. Mutations in the genetic code. Mutations in the genetic code can alter the sequence of amino acids in a protein, which can have serious consequences. Mutations are classified into several types. Point mutations, a change in a single nucleotide, which can result in a silent mutation, no change in the amino acid, a missense mutation, a change in the amino acid, or a nonsense mutation, a stop codon is introduced prematurely. Frameshift mutations, insertions or deletions of nucleotides that change the reading frame of the codons. Previous year questions PYQs 1, CSIR Net Life Sciences 2016. The genetic code is often described as being degenerate. What does this mean? Each amino acid is encoded by a single codon. B. Each codon codes for multiple amino acids. C. Multiple codons can code for the same amino acid. D. The genetic code is read in overlapping triplets. Answer. C. Multiple codons can code for the same amino acid. Explanation. The degeneracy of the genetic code means that there are multiple codons for most amino acids. For example, serine can be encoded by UCU, UCC, UCA, or UCG. 2. CSIR Net Life Sciences 2017, which of the following is true regarding the genetic code? A. It is universal in all organisms, including viruses. B. It is degenerate, meaning one codon codes for multiple amino acids. C. It is non-overlapping, meaning each nucleotide is part of only one codon. D. It is the same in mitochondria and the cytoplasm of eukaryotic cells. Answer. C. It is non-overlapping, meaning each nucleotide is part of only one codon. Explanation. The genetic code is non-overlapping, meaning each nucleotide belongs to only one codon. It is degenerate, but not all codons code for the same amino acid. 3. CSIR Net Life Sciences 2018, which codon acts as a start codon in translation? A. UGA. B. UAA circa August D. UAG. Answer. C. August. Explanation. August is a start codon in translation, encoding the amino acid methionine in eukaryotes and formal methionine in prokaryotes. This codon initiates protein synthesis. Conclusion. The genetic code is a cornerstone concept in molecular biology, providing the blueprint for protein synthesis. Its redundancy, non-overlapping nature, and universality are key features that allow cells to efficiently translate nucleotide sequences into functional proteins. Understanding the nuances of the genetic code and its exceptions is essential for CSIR Net JRF Life Sciences preparation, as it forms the basis for many molecular biology processes. You said. Amino acylation of Turna. ChatGPT said. Amino acylation of Turna. CSIR Net JRF Life Sciences. Maximum detail. Paragraph format. No tables. PYQs included with year and answers. Amino acylation of Turna, also known as Turna charging, is a critical process in protein synthesis. This process ensures that the correct amino acid is attached to its corresponding transfer RNA Turna molecule, enabling the Turna to deliver the amino acid to the growing polypeptide chain during translation. Amino acylation is catalyzed by enzymes known as amino acyl Turna synthetases, and this step is essential for the accuracy and efficiency of the translation process. 1. Role of Turna in translation. Transfer RNA Turna serves as a molecular adapter between the genetic code carried by mRNA and the amino acids that are incorporated into the growing polypeptide chain. Each Turna molecule has an anticodon region that recognizes and binds to a complementary codon on the mRNA and an acceptor stem where an amino acid is attached. 2. Amino acyl Turna synthetases, the key enzymes. 1. Role of Turna in translation. Transfer RNA Turna serves as a molecular adapter between the genetic code carried by mRNA and the amino acids that are incorporated into the growing polypeptide chain. Each Turna molecule has an anticodon region that recognizes and binds to a complementary codon on the mRNA and an acceptor stem where an amino acid is attached. 2. Amino acyl Turna synthetases, the key enzymes. The process of amino acylation begins with the amino acyl Turna synthetase enzymes, of which there are at least 20 distinct types, one for each of the 20 standard amino acids. Each amino acyl Turna synthetase is highly specific for its corresponding amino acid in Turna. These enzymes ensure that the correct amino acid is attached to its matching Turna, which is crucial for the fidelity of protein synthesis. Of amino acylation reaction, the amino acylation process occurs in two main steps. Activation of the amino acid. The enzyme amino acyl Turna synthetase first binds to its corresponding amino acid in ATP, adenosine triphosphate. The ATP is hydrolyzed to AMP, adenosine monophosphate, and pyrophosphate P. The ATP is hydrolyzed to AMP, adenosine monophosphate, and pyrophosphate PPI, activating the amino acid. The activated amino acid is then covalently attached to the adenine residue of the Turna's acceptor stem, forming an amino acyl AMP intermediate. Transfer of the amino acid to Turna. The amino acid is transferred from the amino acyl AMP intermediate to the 3' end of the Turna's acceptor stem, specifically to the hydroxyl group O of the adenosine residue. This results in the formation of an amino acyl Turna complex. The Turna is now charged and ready to participate in translation by delivering its amino acid to the ribosome. 3. Accuracy and proofreading mechanisms. Given that the correct incorporation of amino acids is vital for protein function, amino acyl Turna synthetases possess proofreading activities to ensure accuracy. There are two main proofreading mechanisms. Exonuclease activity. After attaching the amino acid to the Turna, if an incorrect amino acid is attached, the amino acyl Turna synthetase can hydrolyze the incorrect bond using its exonuclease activity and replace it with the correct one. Editing sites. Some amino acyl Turna synthetases. Amino acylation of Turna, also known as Turna charging, is a critical process in protein synthesis. This process ensures that the correct amino acid is attached to its corresponding transfer RNA Turna molecule, enabling the Turna to deliver the amino acid to the growing polypeptide chain during translation. Amino acylation is catalyzed by enzymes known as amino acyl Turna synthetases, and this step is essential for the accuracy and efficiency of the translation process. 1. Role of Turna in translation. Transfer RNA Turna serves as a molecular adapter between the genetic code carried by mRNA and the amino acids that are incorporated into the growing polypeptide chain. Each Turna molecule has an anticodon region that recognizes and binds to a complementary codon on the mRNA and an acceptor stem where an amino acid is attached. 2. Amino acyl Turna synthetases, the key enzymes. The process of amino acylation begins with the amino acyl ternus synthetase enzymes, of which there are at least 20 distinct types, one for each of the 20 standard amino acids. Each amino acyl ternus synthetase is highly specific for its corresponding amino acid and terna. These enzymes ensure that the correct amino acid is attached to its matching terna, which is crucial for the fidelity of protein synthesis. Of amino acylation reaction, the amino acylation process occurs in two main steps. Activation of the amino acid. The enzyme amino acyl ternus synthetase first binds to its corresponding amino acid and ATP, adenosine triphosphate. The ATP is hydrolyzed to AMP, adenosine monophosphate, and pyrophosphate PPI, activating the amino acid. The activated amino acid is then covalently attached to the adenine residue of the ternus acceptor stem, forming an amino acyl AMP intermediate. Transfer of the amino acid to terna. The amino acid is transferred from the amino acyl AMP intermediate to the 3' end of the ternus acceptor stem, specifically to the hydroxyl group O of the adenosine residue. This results in the formation of an amino acyl terna complex. The terna is now charged and ready to participate in translation by delivering its amino acid to the ribosome. 3. Accuracy and proofreading mechanisms. Given that the correct incorporation of amino acids is vital for protein function, amino acyl ternus synthetases possess proofreading activities to ensure accuracy. There are two main proofreading mechanisms. Exonuclease activity. After attaching the amino acid to the terna, if an incorrect amino acid is attached, the amino acyl ternus synthetase can hydrolyze the incorrect bond using its exonuclease activity and replace it with the correct one. Editing sites. Some amino acyl ternus synthetases possess editing sites that further proofread the attached amino acid, removing misacylated ternus before they enter the translation process. 4. Significance of amino acylation in protein synthesis. Amino acylation is a crucial step because the correct charging of terna ensures that the genetic code is faithfully translated into a corresponding protein sequence. Any error in this process can result in misincorporation of amino acids, leading to the production of dysfunctional or non-functional proteins, which may have severe consequences for the cell. 5. Amino acylation in the ribosome. Once terna is charged with its respective amino acid, it is ready to participate in the translation process within the ribosome. The amino acyl terna binds to the A site of the ribosome, where its anticodon binds to the codon on the mRNA. This interaction ensures that the correct amino acid is incorporated into the growing polypeptide chain, according to the sequence encoded by the mRNA. 6. Relationship between amino acylation and translation initiation. Amino acylation occurs prior to translation, preparing terna molecules for the elongation phase of translation. During translation initiation, the ribosome assembles on the mRNA, and initiator terna charged with methionine in eukaryotes or formylmethionine in prokaryotes binds to the start codon of the mRNA. As the ribosome moves along the mRNA, amino acyl ternas enter the ribosome site, where their amino acids are added to the growing polypeptide chain during elongation. Previous year questions PYQs 1. CSIR Net Life Sciences 2016. Which of the following is the enzyme responsible for the amino acylation of terna? Of amino acyl ternus synthetase. bRNA polymerase cDNA ligase depeptidyltransferase. Answer. Of amino acyl ternus synthetase. Explanation. Amino acyl ternus synthetase is the enzyme responsible for attaching the correct amino acid to its corresponding terna, a process known as amino acylation. 2. CSIR Net Life Sciences 2018. Which of the following is true about the amino acylation of terna? A. It occurs in the cytoplasm of prokaryotes and eukaryotes. B. It occurs only in the mitochondria. C. It is carried out by DNA polymerase. D. The process involves the use of GTP for energy. Answer. A. It occurs in the cytoplasm of prokaryotes and eukaryotes. Explanation. Amino acylation occurs in the cytoplasm, where amino acyl ternus synthetase enzymes facilitate the attachment of amino acids to terna. This step is essential for protein synthesis in both prokaryotic and eukaryotic cells. 3. CSIR Net Life Sciences 2017. What is the role of ATP in the amino acylation process? A. ATP is used to hydrolyze the terna during amino acylation. B. ATP is used to activate the amino acid, making it capable of attaching to terna. C. ATP is involved in proofreading the amino acylated terna. D. ATP provides the energy for ribosomal movement during translation. Answer. B. ATP is used to activate the amino acid, making it capable of attaching to terna. Explanation. During amino acylation, ATP is hydrolyzed to activate the amino acid, enabling it to form a bond with the terna. This energy is necessary for the correct charging of the terna. Conclusion. The amino acylation of terna is an essential biochemical process that ensures the fidelity of protein synthesis. By charging terna with the correct amino acid, amino acyl ternus synthetases enable the accurate translation of the genetic code into functional proteins. Understanding this process is key to comprehending the molecular machinery of translation and its regulation, making it a critical topic for CSIR Net JRF Life Sciences preparation. You said. Terna identity. ChatGPT said. Terna identity and its role in translation. CSIR Net JRF Life Sciences. Maximum detail. Paragraph format. No tables. PYQs included with year and answers. Terna identity refers to the specific recognition between a terna molecule and the amino acyl ternus synthetase that charges it with the correct amino acid. This recognition is essential for ensuring the fidelity of protein synthesis, as it ensures that the correct amino acid is added to the growing polypeptide chain in response to the codons on mRNA. The process of terna identity is determined by a set of unique structural features of ternas that allow them to be specifically recognized by their corresponding amino acyl ternus synthetases. 1. Structural features determining terna identity. Each terna molecule has a specific anticodon loop and acceptor stem that interact with amino acyl ternus synthetases, enabling the correct amino acid to be attached. However, the identity of terna is not solely determined by the anticodon region. Several other structural features also play a crucial role in determining the terna's identity. Anticodon sequence. The anticodon of the terna directly pairs with the complementary mRNA codon during translation. While the anticodon is important for codon recognition, it does not directly contribute to amino acylation specificity. Acceptor stem. The 3' end of the terna, which carries the attached amino acid, is essential for the recognition of the correct terna by amino acyl ternus synthetases. T-arm and D-arm. These regions of the terna, consisting of conserved bases, are critical for the proper folding and structure of the terna, influencing its interaction with the enzyme. Other modifications. Post-transcriptional modifications of certain nucleotides in the terna, such as pseudouridine and dihydrouridine, can play a significant role in maintaining terna identity and influencing the recognition by amino acyl ternus synthetases. 2. Mechanism of terna identity recognition. The process of terna identity recognition involves the interaction between the terna molecule and its corresponding amino acyl ternus synthetase. Each amino acyl ternus synthetase has a high degree of specificity for its corresponding terna, and this specificity is dictated by the recognition of certain sequence motifs and structural features in the terna. The anticodon loop, the acceptor stem, and the extra-arm regions are the primary determinants of this specificity. Amino acyl ternus synthetases are capable of distinguishing between correct and incorrect ternas through multiple recognition points, ensuring that each amino acid is attached to the correct terna. This specificity is vital for the fidelity of protein synthesis, as errors in terna charging could lead to the incorporation of the wrong amino acid during translation, potentially resulting in malfunctioning proteins. 3. Terna identity and amino acylation fidelity. The identity of the terna is critical for maintaining the fidelity of amino acylation. The terna must be correctly charged with its corresponding amino acid to ensure accurate translation. Amino acyl ternus synthetases are responsible for ensuring that this happens with high accuracy, using a combination of structural features and proofreading mechanisms. In some cases, incorrect amino acids can be misattached to terna, but proofreading mechanisms within the synthetase enzymes can correct these errors. Moreover, editing sites in the amino acyl ternus synthetases further ensure that mischarged ternas are rejected before they can participate in protein synthesis. This proofreading function is a vital component of the system that maintains the genetic code's integrity. 4. Terna identity and translational efficiency. In addition to its role in fidelity, the identity of terna also influences the efficiency of translation. Codon-anticodon interactions between the mRNA codons and terna anticodons dictate the speed of translation elongation. In some cases, terna identity can also affect ribosome pausing, recoding events, or the incorporation of non-standard amino acids in response to rare codons. This can influence the speed and accuracy of protein synthesis. 5. Mechanisms for evolution of terna identity. In prokaryotes and eukaryotes, terna identity has evolved to match the amino acyl ternus synthetases of their respective organisms. Evolutionary modifications in tRNA structure and identity can lead to changes in the genetic code, allowing organisms to adapt to different environmental conditions or incorporate novel amino acids into proteins. For example, some tRNA synthetases can recognize non-canonical tRNAs, leading to the incorporation of unusual amino acids in specialized proteins. 6. Importance of tRNA identity in genetic code expansion. The identity of tRNA is also relevant in the context of genetic code expansion. Some organisms have developed systems that utilize non-standard amino acids in protein synthesis, often by evolving new tRNA molecules that recognize and incorporate these novel amino acids. This process is crucial in biotechnology, where artificial amino acids are incorporated into proteins for specialized applications in drug design and biomaterial synthesis. Previous year questions. PYQs 1. CSIR Net Life Sciences 2015. Which of the following is directly involved in tRNA identity recognition? A. Ribosome. B. Amino acyl tRNA synthetase cRNA polymerase ddNA polymerase. Answer. B. Amino acyl tRNA synthetase. Explanation. Amino acyl tRNA synthetases are the enzymes responsible for recognizing the identity of tRNA molecules and charging them with the appropriate amino acid, ensuring correct translation during protein synthesis. 2. CSIR Net Life Sciences 2016. Which part of the tRNA molecule is mainly responsible for recognition by the amino acyl tRNA synthetase? A. Anticodon loop. B. Acceptor stem CD loop DT loop. Answer. B. Acceptor stem. Explanation. The acceptor stem is the region of the tRNA molecule where the amino acid is attached, and it plays a critical role in the recognition of tRNA by its corresponding amino acyl tRNA synthetase. 3. CSIR Net Life Sciences 2019. In tRNA identity recognition, the anticodon plays a key role in ensuring the correct amino acid is added to the growing polypeptide chain. B. Identifying the start codon during translation initiation. C. Providing the correct recognition site for the amino acyl tRNA synthetase. D. Matching the tRNA to the corresponding mRNA codon. Answer. D. Matching the tRNA to the corresponding mRNA codon. Explanation. The anticodon of tRNA specifically recognizes and pairs with the codon on the mRNA during translation, ensuring that the correct amino acid is incorporated into the growing polypeptide chain. Conclusion. tRNA identity is a fundamental concept in molecular biology and protein synthesis. It ensures that the correct amino acid is linked to the appropriate tRNA, thereby maintaining the accuracy of translation. The specific recognition between the amino acyl tRNA synthetase and its tRNA substrates is central to protein synthesis fidelity, and understanding this process is essential for cracking the CSIR Net JRF Life Sciences exam. The mechanisms that regulate and maintain tRNA identity are crucial for the translation process and are vital for cellular function. You said. Amino acyl tRNA synthetase. ChatGPT said. Amino acyl tRNA synthetase. Structure, function, and mechanism. CSIR Net JRF Life Sciences. Maximum detail. Paragraph format. No tables. PYQs included with year and answers. Amino acyl tRNA synthetases (AARSs) are a group of enzymes that play a pivotal role in protein synthesis by charging tRNA molecules with their corresponding amino acids, a process that is essential for accurate translation of the genetic code. These enzymes ensure the fidelity of translation by selecting the correct amino acid and attaching it to the corresponding tRNA in a process called aminoacylation. Without this crucial step, the translation machinery would not be able to build proteins correctly, leading to severe cellular dysfunction. 1. Structure of amino acyl tRNA synthetases. Amino acyl tRNA synthetases are highly specific enzymes that recognize both the amino acid and the tRNA molecule. These enzymes typically consist of several functional domains, which include catalytic domain, the active site where the amino acid is attached to the tRNA molecule. It is responsible for the ATP-dependent charging of tRNA with its corresponding amino acid. Editing domain. Many synthetases possess an editing site that allows the removal of incorrectly attached amino acids, providing a proofreading function to maintain fidelity. tRNA recognition domain. This domain helps the synthetase recognize the specific structural features of the tRNA that correspond to a particular amino acid. Attachment domain. In some synthetases, a domain is involved in the physical attachment of the tRNA to the enzyme. There are two main classes of amino acyl tRNA synthetases based on their structural characteristics and the type of catalytic mechanism they employ, class 1 and class 2. Class 1 amino acyl tRNA synthetases generally recognize the 3' end of the tRNA, where the amino acid is attached, and they tend to have larger, more complex structures. Class 2 amino acyl tRNA synthetases recognize the acceptor stem and have a simpler architecture compared to class 1. 2. Function of amino acyl tRNA synthetases in protein synthesis. Amino acyl tRNA synthetases are critical for translating the genetic code from nucleic acids to proteins by charging tRNAs with their corresponding amino acids. This step ensures that the appropriate amino acid is incorporated into the growing polypeptide chain during translation. Each amino acyl tRNA synthetase is specific to a single amino acid and its corresponding set of tRNAs, and it performs the following key functions. Amino acid activation. The enzyme first binds to the amino acid and uses energy from ATP hydrolysis to activate the amino acid, forming an amino acyl AMP intermediate. Amino acylation. The activated amino acid is then transferred from the amino acyl AMP intermediate to the 3' end of the tRNA, forming the amino acyl tRNA complex. Proofreading and editing. To ensure the accuracy of the charging process, most amino acyl tRNA synthetases possess an editing function that hydrolyzes incorrectly charged amino acyl tRNAs, ensuring the correct amino acid is attached to the tRNA. 3. Mechanism of amino acylation by amino acyl tRNA synthetase. The process of amino acylation can be broken down into several steps. Step 1. Amino acid binding. The amino acid binds to the active site of the amino acyl tRNA synthetase. The enzyme also binds to ATP, which is required for the activation of the amino acid. Step 2. Activation of amino acid. The enzyme catalyzes the hydrolysis of ATP, forming amino acyl AMP. This intermediate is highly reactive and allows the transfer of the amino acid to the tRNA. Step 3. tRNA binding. The enzyme binds to its corresponding tRNA, specifically recognizing the anticodon loop and acceptor stem. The tRNA is aligned in such a way that the 3' end of the tRNA is positioned near the amino acyl AMP intermediate. Step 4. Amino acylation of tRNA. The activated amino acid is transferred from amino acyl AMP to the 3' hydroxyl group of the tRNA's acceptor stem, resulting in the formation of an amino acyl tRNA complex. Step 5. Proofreading. If an incorrect amino acid is attached, the editing site of the amino acyl tRNA synthetase removes the mischarged tRNA, ensuring that only the correct amino acyl tRNA enters the translation machinery. 4. Specificity of amino acyl tRNA synthetases. The specificity of amino acyl tRNA synthetases is fundamental for the correct translation of the genetic code. Each enzyme is highly specific not only for its amino acid but also for the tRNA molecules that correspond to that amino acid. This specificity is conferred by several mechanisms. Recognition of the tRNA structure. The synthetase interacts with conserved structural features of the tRNA molecule, including the anticodon loop, acceptor stem, and extra arm regions. Structural complementarity. The enzyme's active site fits precisely with the amino acid in tRNA, ensuring that the correct amino acid is attached to the appropriate tRNA. Proofreading mechanisms. Many amino acyl tRNA synthetases contain editing sites that allow them to correct any errors in amino acylation, adding another layer of specificity to the process. 5. Role of amino acyl tRNA synthetases in genetic code expansion. Amino acyl tRNA synthetases are not only involved in the translation of the standard genetic code but also in the expansion of the genetic code in certain organisms. For example, synthetases can be engineered to incorporate non-standard amino acids into proteins, a process that is widely used in synthetic biology. This ability to modify the genetic code holds great potential for the development of new proteins with novel properties for applications in drug design, biomaterials, and therapeutics. Previous year questions. PYQs 1. CSIR Net Life Sciences 2016. Which of the following enzymes is directly responsible for attaching amino acids to the tRNA molecule during protein synthesis? A. DNA polymerase. B. RNA polymerase. C. Amino acyl tRNA synthetase. D. Ribosome. Answer. C. Amino acyl tRNA synthetase. Explanation. Amino acyl tRNA synthetases are responsible for attaching the appropriate amino acid to its corresponding tRNA molecule during protein synthesis. 2. CSIR Net Life Sciences 2018. Which of the following is the primary function of the editing domain in amino acyl tRNA synthetases? A. To attach amino acids to the tRNA. B. To correct errors in amino acylation. C. To catalyze the hydrolysis of ATP to bind to the tRNA anticodon. Answer. B. To correct errors in amino acylation. Explanation. The editing domain of amino acid synthetases plays a crucial role in proofreading the amino acylation process by hydrolyzing mischarged ternus, ensuring the accuracy of protein synthesis. 3. CSIR-NET Life Sciences 2020. Which of the following functional domains is essential for the attachment of the correct amino acid to the ternus in amino acyl ternus synthetases? A. Editing domain. B. Catalytic domain. C. Anticodon recognition domain. D. ATP binding domain. Answer. B. Catalytic domain. Explanation. The catalytic domain of amino acyl ternus synthetases is responsible for the attachment of the correct amino acid to the ternus molecule, a key step in protein synthesis. Conclusion. Amino acyl ternus synthetases are integral to the fidelity of protein synthesis by ensuring the correct amino acid is attached to its corresponding ternus. Their specificity and proofreading mechanisms are critical for maintaining the integrity of the genetic code. Understanding the structure, function, and mechanisms of these enzymes is essential for cracking the CSIR-NET JRF life sciences exam, and it provides insights into the molecular machinery that underpins cellular life. You said. Translational proofreading. ChatGPT said. Translational proofreading. Mechanisms and importance in protein synthesis. CSIR-NET JRF life sciences. Maximum detail. Paragraph format. Note tables. PYQs included with year and answers. Translational proofreading is a crucial process that ensures the fidelity of protein synthesis by minimizing errors in the translation of the genetic code into proteins. During translation, ribosomes read the mRNA sequence and synthesize proteins by incorporating the correct amino acids into the growing polypeptide chain. However, the translation process is not flawless, and errors can occur, leading to the incorporation of incorrect amino acids, which can result in malfunctioning proteins and potentially disastrous cellular consequences. Translational proofreading involves multiple layers of error-checking mechanisms to maintain accuracy, such as amino acyl ternus selection, peptide bond formation, and termination. 1. Role of amino acyl ternus synthetase in proofreading. The first layer of proofreading and translation occurs during aminoacylation by amino acyl ternus synthetases. These enzymes attach the correct amino acid to its corresponding ternus, a process that is critical for accurate translation. To ensure the accuracy of this process, amino acyl ternus synthetases possess editing sites that can remove incorrectly attached amino acids. This proofreading function is essential because even a single incorrect amino acylation can result in the incorporation of the wrong amino acid during translation. Initial charging. In the first step, the synthetase attaches the amino acid to the ternus. Editing mechanism. If the wrong amino acid is charged to the ternus, the enzyme can hydrolyze the mischarged ternus, preventing its entry into the ribosome. This editing step ensures that only the correctly charged ternus enters the ribosome for translation. This proofreading by amino acyl ternus synthetases is highly efficient and occurs before the ternus enters the ribosome, making it the first line of defense against errors in translation. 2. Proofreading mechanisms at the ribosome. Once the correctly charged ternus enters the ribosome, further proofreading mechanisms are involved in ensuring that the correct amino acid is incorporated into the polypeptide chain. These mechanisms are largely dependent on the structure and function of the ribosome. Codon-anticodon interaction. The ribosome reads the mRNA sequence in triplets codons, each of which corresponds to an amino acid. The anticodon of the ternus pairs with the codon of the mRNA. This interaction is critical for ensuring that the correct ternus is selected for the correct codon. Selective elongation. Once the ternus is in place, the ribosome facilitates peptide bond formation. If the ternus anticodon does not match the mRNA codon with high fidelity, the elongation process is delayed, and the incorrect ternus is rejected from the ribosome. This process acts as a second proofreading step, ensuring that only the ternus carrying the correct amino acid is incorporated into the growing protein. 3. Mechanisms of proofreading and elongation factors. In addition to the amino acyl ternus synthetase in the ribosome, the elongation factors such as EF2 in prokaryotes and EF1A in eukaryotes play a crucial role in translational proofreading. GTP hydrolysis. Elongation factors help deliver the charged ternus to the ribosome. The elongation factor bound to GTP first ensures that the ternus is correctly matched to the mRNA codon. Once the ternus is properly positioned, the elongation factor undergoes GTP hydrolysis, which changes its shape and releases the ternus into the ribosome. If the ternus is not correctly matched with the codon, the elongation factor will prevent its release and block the peptide bond formation. Proofreading by elongation factors. This GTP hydrolysis-based proofreading ensures that only the correct ternus is incorporated into the growing protein chain. 4. Peptide bond formation and proofreading. Peptide bond formation is another critical step where proofreading occurs. After the ternus is positioned correctly in the ribosome, the peptidyl-transferase center catalyzes the formation of a peptide bond between the growing polypeptide chain and the new amino acid. If an incorrect ternus has been incorporated, the peptide bond formation is hindered. This proofreading mechanism ensures that the amino acid sequence remains accurate during protein synthesis. 5. Termination and proofreading. The termination phase also involves proofreading to ensure that the protein synthesis process stops at the correct point. Release factors such as RF1, RF2, and RF3 in prokaryotes and ERF in eukaryotes are responsible for recognizing the stop codon in the mRNA and catalyzing the release of the finished polypeptide chain. If the translation machinery encounters a nonsense mutation, a premature stop codon, the proofreading mechanisms involved in ribosome dissociation and release factor activation ensure that translation terminates at the correct location. 6. Proofreading and quality control mechanisms. Besides the direct proofreading steps in the translation process, there are quality control mechanisms in place to handle errors in translation. One such mechanism is the translational fidelity checkpoint, which involves the surveillance systems like non-stop decay (NSD) and no-go decay (NGD) pathways. These systems detect and resolve errors such as stalled ribosomes or frame shift errors during translation. Previous year questions. PYQs 1. CSIR-NET Life Sciences 2017. Which of the following factors contributes directly to ensuring the correct codon-anticodon matching during translation? Of amino acyl ternus synthetase. B. Elongation factor G. EFG. C. Ribosome associated release factor D. Peptidyltransferase activity. Answer. Of amino acyl ternus synthetase. Explanation. Amino acyl ternus synthetase ensures the correct amino acid is attached to the ternus, which directly affects the codon-anticodon matching during translation. 2. CSIR-NET Life Sciences 2019. What is the primary role of elongation factors in translational proofreading? A. To charge ternus. B. To ensure the correct codon-anticodon matching. C. To catalyze peptide bond formatio.